Good afternoon, colleagues, friends, and relatives. On behalf of Vice Chancellor and Principal Professor Nana Poku, it gives me a great pleasure to welcome you to the inaugural lecture of Professor Samson Zeray Tesfay. The Vice Chancellor conveys his congratulations and best wishes to Professor Samson Tesfay, and I would like to acknowledge the following guests this afternoon: the members of Council, members of Executive Management, members of Senate, our inaugurant this afternoon, Professor Samson Zeray Tesfay, family and friends of Professor Tesfay, academics, professional staff, students, alumni, and distinguished guests. A special welcome to our guests from universities and organizations within South Africa, the African continent, and across the globe. Just to give you a little bit of a background of our inaugural lectures. Inaugural lectures form part of the University Public Lecture Series and may only be presented by newly appointed full professors who have been appointed in academic schools and centers. Inaugural lectures presents an opportunity for our showcasing the exciting and groundbreaking research and teaching being carried out by professors in our university. Each lecture represents a significant milestone in an academic career, providing official recognitions of their promotion or appointment to full professorship. And these lectures are furthermore an ideal opportunity for our new professors to introduce themselves and to present an overview of their own contribution to their field to academic peers, students, and research collaborators. Inaugural lectures are also a platform of celebrating a professor's academic achievement with his, her family, friends, mentors, and colleagues. Distinguished guests, it is my pleasure to now introduce the Dean and Head of School of Agriculture and Science, Professor Ademola Olaniran, who will now formally introduce the inaugurant, Professor Samson Zeray Tesfay. Over to you, Prof.
Thank you. Good afternoon, everyone. It is with great honor that I introduce our inaugurant for this afternoon, Professor Samson Tesfay, who is a full professor in horticultural sciences within the School of Agriculture and Science, within the College of Agric Engineering and Science, University of KwaZulu-Natal in South Africa. Professor Tesfay received his BSc Agric Arid Zone Crop Production from the University of Asmara, Eritrea in 1994. He obtained his MSc and PhD degrees in crop science and horticultural sciences in 2006 and 2010, respectively, from the University of KwaZulu-Natal. He received the IMF scholarship for his MSc program and South African Avocado Growers Association scholarship for his PhD study. In his early careers, Professor Tesfay worked as Extension Officer and HOD for the Department of Agriculture in Eritrea. He then joined the Department of Horticulture at the University of KwaZulu-Natal in 2013 as a lecturer. He further ascended to the position of Senior Lecturer in 2018, Associate Professor in 2020, and Full Professor in 2024. In addition to his academic achievements, his early career experience as an Extension Officer and Professional Technical Director in Agronomy Division in Zone Anzeba, Eritrea, provided him with a solid foundation in practical agriculture, which has informed his teaching and research. This unique blend of practical and theoretical knowledge positions him to effectively contribute to the discipline of horticultural sciences, particularly in bridging the gap between research and application in the field of horticulture. Professor Tesfay's research is in the area of plant secondary metabolites and post-harvest fruit physiology. In particular, he works extensively in plant antioxidants, fruit tissue antioxidant distribution, and their uses towards fruit quality attributes. The main focus of his research in plant antioxidants is the notion of antioxidant functions, the extraction methods, and its various uses as pre-harvest and post-harvest treatment. Professor Tesfay has successfully completed the supervision of 12 doctoral and 35 master students. He has also mentored 6 postdoctoral fellows, and he has published over 160 research articles in Scopus and ISI index journals over the years. He is currently supervising 5 doctoral students within the University of KwaZulu-Natal. As a full professor in horticultural sciences at the University of KwaZulu-Natal, with over a decade of academic and practical experience in horticultural sciences, along with passion for advancing research in plant secondary metabolites and post-harvest technophysiology, Professor Tesfay has developed a robust research portfolio that encompasses the investigation of plant antioxidants and their significant role in enhancing food quality attributes. This includes his work on antioxidant functions, extraction methods, and the application of these substances as both pre-harvest and post-harvest treatment. Professor Tesfay has examined a number of master's and PhD theses from various universities, including University of Johannesburg, Stellenbosch University, University of Pretoria, Tshwane University of Technology, Durban University of Technology, University of Limpopo, University of Fort Hare, UNISA, Nelson Mandela University, and universities from the United Arab Emirates. He is a member of the editorial board of the International Society of Horticultural Sciences. He is also a reviewer for more than 20 high-impact faculty journals in his research field, including Scientia Horticulturae, Post-Harvest Biology and Technology, Plants, Australian Journal of Botany, New Zealand Journal of Crop and Horticultural Science, Frontiers in Plant Science, Food Packaging and Shelf Life, Food and Bioprocess Technology. Professor Tesfay is a C2 rated scientist by the National Research Foundation of South Africa, and this rating he has since 2023. He has been one of the top 30 productive researchers in Africa in his area of specialization. He is a member of South African Fruit Growers Association, South African Macadamia Growers Association, and the International Society of Horticultural Science. Professor Tesfay, congratulations on your academic achievement. And may I please invite you to deliver your inaugural lecture titled Rethinking Plant-Based Secondary Metabolites in Fruits, Tissue Distribution, and Byproduct/Food Waste Valorization for Environmental Sustainability.
Thank you, Paul. Thank you for that introduction. Good afternoon, dear Prof. Midal, DVC of the College of Agriculture, Engineering and Science, dear Prof. Olarina, Dean of the School of Agriculture and Science, dear colleagues, family and friends. I'm deeply grateful to each of you for taking the time to attend my inaugural lecture celebrating my full professorship. It is an honor to have your presence and support in this significant occasion. So before embarking on today's lecture on antioxidants, I would like to extend my heartfelt thanks to the following people and my family members who have been instrumental in this journey. My wife, Nonkuleko Tesfay, my kids, Lorenzo, Lemuel, Asema, and also my parents. My mom is here watching, very proud. And then all my siblings and a close family who are attending online. Thank you so much for your unwavering support and belief in me, and which has been my anchor. And secondly, Prof. Ndijwa, Prof. Magwaza, Prof. Mabauti, Prof. Odindo, thank you for your friendship all this time, all these years. And then I appreciate your continuous motivation and for being a sounding board for my ideas in plant antioxidants. Also, I would like to thank Prof. Bertley, Prof. Bauer, Prof. Greenfield. Thank you for your support. And then allow me to work with you as a student, as well as a colleague. And please be aware that many of you who are present here have played significant roles in one way or another. Although I haven't named all of you on this occasion during the lecture, you should know that I have interacted with you at many levels, anytime. Your support and contribution have not gone unnoticed. I'm truly grateful for that. You believe in my capabilities and your constant encouragement have been pivotal in achieving this beautiful milestone. I'm truly fortunate to be surrounded by such a supportive community at large. Thank you once again for being here today. I look forward to share my insights on the secondary metabolites and then engaging with you during the lecture. So I just introduced my families. This is my family members, my family from England, my siblings, and then also that's all my families also from here. Then by saying that, I would like to introduce you to today's topic, which I decided to do on rethinking, so reconsidering the food waste, mainly on the plant-based secondary metabolites in foods, tissue distribution in plant byproducts, valorization for environmental sustainability. So my focus is going to be on firstly, to look at the plant metabolites biosynthesis, and then also the mode of action at cellular level. And then we look also at the different types of extraction methodologies. Well, and then with this metabolites also, how do they distribute in plant tissues in different parts of plant organs? And then I'll move to what do we do with the extracts? Currently, as one of the case studies we do in our research, we use the extracts for post-harvest treatments because of the fact that they are dominant in secondary metabolites. And then we use the same extract for antimicrobial, as antimicrobial agent, as this is very critical as it's considered as an alternative to synthetic fungicides. Lastly, we also embark into the nanotechnology, which we believe that this is the future, the game changer in terms of delivery of those metabolites. So with the graphical outlines, here simply a follow-up of the outline, which is food waste recycling and processing, and dealing with the fruits and vegetables, the fresh produce. Either you can go the other pathways, which is processing, but our focus will be here on how do we deal with food waste. With food waste, then we actually use different extraction protocols, and then also valorization, dealing with the extracts. Then that includes characterizing them, identifying, and purification. Then also we go for bioactivity. What are the properties they have as an antioxidant as well as antimicrobial? But for consumers, they also benefit for the health benefits. And then we use the extracts also, we can also form different forms of the products for functional foods that can be also valorization or adding values where it actually contributes for sustainable agriculture. So to start with, let's see what is the current perspective of agriculture. As you're all familiar with Green Revolution, where agricultural productivity has increased dramatically in the last 60 years. However, this approach, although they allow usage of the heavy or intense agrochemicals, that resulted with a number of challenges to the environment that creates environmental degradation, climate change, and then lastly, I will highlight on the poor soil health. And also the rate of the crop yield increase has declined since the 1980s, as this is attributed to the climate change and environmental degradation. As a result, global food security is existed and then also aggravated by malnutrition. People are chronically hungry in some parts of the world, suffer also from such cause. So this challenge basically will actually take us to the dire need for sustainable development, which is sustainable agriculture. We're tapping on the plant byproducts, which we consider is a plant waste or plant byproducts. Then why plant secondary metabolite is as a candidate for sustainable agriculture? These are the motives. The first motive is the global population is expected to increase to 9.7 billion in 2050. This obviously leads to further increase in plant waste production by 70%. So we have a sort of an opportunity here to reutilize this food waste to valorization. The second motive is the climate change. The climate change dictates the diversity of a number of different species. Those adapted, they are known to accumulate high level of antioxidants. So the secondary metabolites contribute to sustainable agriculture as well. The last motive, if you look at the non-edible portion of the plant byproduct, it accounts from 25 to 30%. So this can also be potentially reutilized. I consider it as an opportunity. And then this non-edible portion also includes the exocap, which is the peel, the seed part, and the leaf. And then also it's reported that these tissues are known for high accumulation of secondary metabolites, antioxidants. And then in addition to that, also this byproducts also they contain non-digestible components, and phytonutrients can also be reutilized and then add values, valorization. So given these motives, so for sustainable agriculture, then we need to adopt innovative strategies that includes recovering plant secondary metabolites from the byproducts, adopting and improving climate-smart crops, and then improving soil health, and then also adopting some bioproducts such as biostimulants, and then nanotechnology will also allow in the efficiency or delivery of those metabolites for agricultural purposes. So with the non-edible portion here, this table demonstrates a list of tables, the list of fruit and vegetables, and then also a comparative proportion of byproducts in edible. So it's given different magnitudes, but the highlight of this slide is that byproducts, the exocap in a seed, they are known to actually give high level of antioxidants compared to the edible part. In fact, the difference is actually 40 times higher than what is actually given, what is obtained in the edible. So there is also a big opportunity to recycle. So now let's look how do the synthesis of these metabolites actually happen at cellular level. In fact, with the secondary metabolites, there is also primary metabolites to be discussed, considered as they are the primary or the starting point for secondary metabolites. So what are these primary metabolites? It includes carbohydrates, lipids, proteins, nucleic acids. They are known also as a source of energy. And then these metabolites also, they are directly involved in plant growth, development, as well as production. So the carbon metabolism at cellular level, it occurs at the carbon cycle. So with this one, I just want to highlight, what are the power of the cellular functions. And then also with the cell structure, if you look at, it's actually intensely composed of these organelles. These organelles also, they have special kinds of tasks and functions. So to be also precise, I want to highlight on the chloroplast. The chloroplasts are the one actually responsible for the absorption of light. We have the energy transfer. Then this energy transfer within the chloroplast, it has to go to the reaction center where the plant photosystems are occur. So with the photosystem, then there will be electron excitations. Those energy excitation actually transported through the electron chain, electron transport chain, and then the amount of electron transport rate actually directly proportional to the amount of organic compounds. So everything that we talk about, primary, secondary metabolites, actually it occurs at cellular level. Then this demonstrates the chlorophyll pigments and different compounds as well. So simply, this is a demonstration of what happened. These are the chlorophylls. And then in fact, the light energy transferred from one chlorophyll to others to get it to the electron center or reaction center. So we have a light spectra here, then a light absorbed by the leaf cellulite chloroplast, which is chlorophyll, and then transferred, and then comes to the reaction center. These electrons actually, they play a huge part in the formation of ATP as well as NDPH. Those are the electron molecules, the electron transport molecules. They will get to the carbon cycle. Then they participate critically on the synthesis of the metabolites. Those metabolites include primary as well as secondary metabolites. So this is a photoreaction. As you can see, there's a photosystem too. The electrons comes out of here, and then here it will get to the point where ATP synthase and then form ATP. And then the water split it because electrons has to be inside photosystem to replace what is already excited or left the photosystem to, then that allows the normal functioning of the cell. So this basically, it ends up in formation of NDPH. So I think with this one, quickly I will pass on. This is in real life. The leaf actually is actively involved in light, and then carbon dioxide in water. And then there's this one, the second one, you can see that as a chloroplast. Then we have chloroplast, chlorophyll pigments, and then also thylakoid, the stroma and lumen. And then that's what the formation of the ATP and NDPH actually formed at cellular level. Then we have also here, if you look at, in fact, in this excited, and then it comes out of here because of the excitement from the light. Then lastly, it will also move to the ADP, and then the formation will happen here. This is in the real sense at cellular level. So let's move to the secondary metabolite. What are these secondary metabolites? They are different from primary, but their production, although it's actually derived from the primary metabolites, but their function is totally different. This is actually in response to adaptation or defense mechanism. So basically, they are important for ecological functions. So since we have lots of actually secondary metabolites, so far around 50,000 is actually reported. But what is important here to understand the distinct pathways, the shikimic, which is responsible to phenols, the mevalonate pathways responsible for the terpenols, and then also the acetate malonate pathways is responsible for the fatty acids or alcohol. So we have primary carbon metabolisms, then you have a number of different actually metabolites, including the secondary metabolites. It's actually happened at the leaf organ, but leaf actually is compacted with different number of cells. So we have the light here, CO2 water. The leaf is the organ is responsible for photosynthesis, and then also production of carbohydrates, which is carbon cycle. The carbon cycle, these are the different pathways which actually allows the formation of nitrogen containing the secondary metabolite, phenolic compounds, and including terpenoids. So with this one, what I want to highlight here, although it's similar to the previous slide, what happens, the plant cell undergoes metabolism. So in one of the biological processes, what happens at the cellular level is respiration. So basically you have the carbohydrates or the energy plus oxygen. Then of course, with that metabolism, then we have this mode of action. Plant cell metabolism under normal circumstances, the radicals, which is a reactive oxygen species, they normally produce, but the plant has the ability naturally to counteract those radicals or reactive oxygen species through the production of antioxidants, which in other words, the secondary metabolite. So there are a number of different types, but the quantity is dictated by the high accumulation of this radicals or reactive oxygen species. So just to highlight, salicylic acid, this is basically from the shikimate, but the reason why I highlighted here, we did some work which is published. So basically, this is actually stimuli or metabolites. They are used for actually produced in response to the pathogenic infections or herpesvirus or outside action. So that this is basically follows the shikimate. But if you look at the shikimate, the first ever product of the shikimate, which is the precursor, is corysmate. So corysmate, if you look at it, it's actually responsible for phytohormones and then also as a secondary metabolite. Then with proline, it is a secondary metabolite, amino acids. They are responsible for, they are actually a molecular marker for drought tolerance. This is actually one of the works we did actually with Professor Shimelis at plant breeding. Then of course, we use proline in order to actually dictate the tolerance of the wheat genotype. Then of course, here, so far, this is actually highly cited in my scholar, in the Google Scholar, which I believe that, this is a very beautiful work. And then, of course, this secondary metabolite can be used for many other agricultural activities. So here, what I wanted actually to share with you, then we have a number of different types of phenolics. And then I also presented the different molecular structures. So the structures, it has something to do with the capacity of the antioxidant. So here, the different types. And then, of course, also with here, what I'm trying to link it with sources of those metabolites. So different fruits and vegetables, including tea, actually responsible for certain types of phenolics. Then, of course, that this is where we have to look at it that way. We look at the plant species, then we know that also they are dense in those beneficial compounds, then it can be reutilized. So here, terpenes, then I decided to choose the hemp cannabis. I'm one of the researchers in this field. We do the agronomy, we do the seed productions, but at the same time also we extract the oils, the terpenes, and which is CBD, cannabinoids, and then tetrahydrocannabinol, as you can see it. Then these are the trichomes, and then rich densely actually with the oil, and then we use that extract basically for cosmetics. So that's part of what we are trying to address in today's lecture. This is our products, and then this is our products, and then in fact some of the names I just give it after my daughter. Then these are my postgraduate students. They fully participated in making it. I also learned from some of the students how to make this cosmetic. But what we are saying that we have a potential to do the valorization or developing products. Then with amino acids and alkaloids, we have a different structural, they are totally different. Then also here, I'm highlighting also, these are the source of those alkaloids from the given food species. So this is for nicotine and then also the caffeine molecular. So then the mode of action of the plant secondary metabolites, which I have already said it, but what happens, plant cell metabolisms, then under normal conditions, actually it produces, if it's oxidized, respiration, any maturity process in metabolic compositional changes, then that tend to produce radicals in the growth naturally. So this production naturally also will be counter-reacted by producing antioxidants. So remember here, how does this production actually increased or stimulated? These are the factors where it actually stimulates the production of this rose, more dangerous organism, which is dangerous to the plant cell. Then the plant has the ability actually to produce the enzymatic as well as the non-enzymatic antioxidants. But if it persists, then becomes it toxic. So remember, we have rose and then antioxidant balance here. But when you come down here, it's imbalanced, which is outweighed by reactive oxygen species. So there is a danger here. So what happens, the plant will suffer from this toxic effect. Finally, it affects, it damages the DNA, and then of course it kills the whole plant. Then some of the actions of the antioxidant polyphenols, rose scavenging, and then we have also antioxidant enzyme induction. We have oxidants, and the first enzyme is dismutase, superoxide dismutase, and then that produces hydrogen peroxide, which is rose reactive active species. Then the catalase should also catalyze it to water and oxygen, where at this condition it becomes safe. So it's also used for metal chelations, and then also it also inhibits some rose forming enzymes. So the elicitations, the defense responses, some expression of genes, enzymes, and then that leads to the production of the secondary metabolites. So then speaking of their production, there are a number of stress factors contributing there for their production. So it could be secondary metabolites, so some enzymes also somehow stimulated. So drought stress is responsible for certain, such as flavonoids, and then epicatechins. Salinity also that's responsible for the production. So this is something that, if we can also control this stress condition, we can easily regulate production of secondary metabolites. But here, the challenge is that, is it possible actually to control it under outdoor production system? So those are the different stresses. And then we have also a number of the drought-resistant mechanisms, avoidance, escape mechanism, tolerance. What I want to share with you here, those physiological responses are mainly dictated by the production of secondary metabolites. They could be enzymatic or non-enzymatic. So you can see that we have some stress factors, and then also production of physiological responses such as stomata closure, and then also increasing of some enzymatic enzymes. And then finally, what happens here, enhancing photosynthesis, drought tolerance, and then yield assurance. So these are the true stress factors that regulate different types of secondary metabolites. We have a number of different types of abiotic stresses, and then we have also a number of different types of biotic stresses. Then this regulates, of course, it's good to have a knowledge of these different types of stresses, but we can also manipulate them for the sake of, in favor of production of secondary metabolites. So this different abiotic stress, the signaling, then which I want to highlight here, when you have reduction in oxidation imbalance, that results with the stress intensity. So The extraction is crucial now. So extraction, what are the methods we are adopting? In fact, in our lab, what we currently using, actually, some of it is conventional, probably that detects, the amount of metabolites actually produced, as well as qualitatively, as well as quantitatively. But here, there are some emerging, which we should be aware of, pulse electric field assisted extraction, enzyme assisted extraction with microwave, ultrasonic. So there are a number of, which we need to reconsider as we are aiming to the valorization. So with this, in fact, we're dealing with the extracts, and then we have also different types of extraction protocols we employ. Then the crude extract, and then the bioassay here. We need to have a chemical assays, antioxidant, which we do it in our labs. And then also that, identification. We use a different chromatographic approach, the TLC, which is the old system. Then we normally actually do the whole process of this slide in our labs. So here, comparison case studies, then ultrasonic, for example, assisted extraction approach, it actually improves 25% of increase in total phenol compared to maceration. Microwave assisted extraction, huge, 30 to 35% of higher carotenoid yield actually produced. Then enzyme assisted, the same thing. So it is crucial and vital to get the optimum or the best extraction protocol. So this is the ultrasonic, and then some of the so with the plant secondary metabolites and distributions, here mango, we did some research on that. So dealing with different types of tissues, with the exocap, and then also the edible part, the mango seed, and then this different types of the mango tissues attributes a different proportion. But if you total it, which is a byproduct from 35 to 60%, it's a waste. So we can actually convert it to the actual extraction of secondary metabolites, which is valorization. But the other thing also, what is interesting, that speaking of secondary metabolites, they significantly produce in the outside, which is in the exocap, as well as in the seed tissue of the fruit. So we need to consider. So this is basically the high concentration antioxidant in the peel as seeds, and then also the distribution, high concentration is actually attributed to the metabolic or UV radiation, which is exposed to the harsh conditions, and then also against herbivores and pathogens, and then also the process of ripening and decay, which is similarly similar to the high metabolism that actually allows the plant actually to counteract the high level of oxidants by producing second. This is some of the published work. Then this is also many years ago, we did some research on the antioxidant levels of the tissue of hass fruit. Then what happened that, we looked at the mesocap and seed and then exocap, and then also different parameters of the total antioxidant, as well as also individual. So when you look at the magnitude, in fact, the seed and then exocap produces 40 times higher than the mesocap. So the greater proportion of the secondary metabolites are produced in seed in the exocap. And then also what we did here, we also did some work on the different types of enzymatic antioxidants. So with the phenolics, and then we did also some work on the distribution, as well as also different types of phenolics, where we looked at the free soluble and bound. And then with the phenolics pre-harvest, but if you look at the distribution of phenols, the trend, in fact, the exocap and the seed and the leaf actually had very high concentration of these phenolics compared to the bottom, which is the mesocap. So I think it's important also to reconsider the byproducts for secondary metabolites. Then one of the post-harvest treatments, we did potassium sulfate application on phenols. Then post-harvest treatments, and then we did some study in certain parameters. So I will highlight on the catalyst activity and lipid. The catalyst activity, enzymatic, and then we found out that the silkate treatments had significant effect compared to the control. But with lipid peroxidation, we do it in the post-harvest because this is a very good indicator for physiological disorders such as chilling injury, so that the more lipid production we read, it means this is highly prone to the chilling injury, which is a physiological disorder. So with silkate, then we had also better result in the significance. So with the catalyst, and then in here, we did follow it also with the expression of the protein. So we did catalyst, Western protein, the SDS page, run it in the gel, and then also that, transfer the proteins to the cellulose paper, and then also brought it with antibodies. And then also we found that also this treated fruits had higher expression compared to the control. So in fact, lastly, we checked also different types of phenolic standard compounds against different antioxidant capacities, ABTase, FRAP, and then ORAC. And then different structures contributes different antioxidant capacity. The other thing also, working on antioxidant assays, it is always important and safe to use more than one, such as ABTase. This is because of the nature of the metabolite. Some are hydrophilic, some are lipophilic. So it's important to have more than one assays. So here we looked at the free soluble and bound phenolics. So the mesocap here, interestingly, it is dominant with free soluble phenolics. But whereas the exocap in seed, if you look at it, it's actually the bound phenolics are far much higher than the free ones. So there were significant differences when it comes to the. So with the moringa, I think we've researched moringa for many years. We are very close to it. In fact, from the seed, we understood the seed viability and quality, and then also that we grew it in our tunnels, in the seedling trays, you can see, and then also that transplanted to the pods, and then also studied a number of things, the nutritional proportion at different parts of the tree, and then also exposing them at different temperature regions to check the production of fish. So the reason why moringa is a good candidate is a smart crop, climate smart, and then also it's rich in nutritional profile and functional properties. And then locally, it's known as drumstick. I think very common in South Africa as well. So in some places they use it as a biostimulant because of its dense in terms of the extract, and then also we used it as a post-harvest treatment for food qualities as well as also the antifungal agent. So looking at the different parameters, different types of the plant parts, then of course that shows that the different parts of the tree actually contributes to different types of compounds. Metabolites includes primary as well as secondary metabolites. So with the membrane-bound phenols, the same thing. So, and then also furtherly, we looked at the leaf extracts, characterize it using different spectrophotometry procedures, gas chromatography, mass spectrophotometry. Then we characterize the different types of compounds. And then also that we looked at the HPLC high performance, liquid chromatography, mass spectrum, and then we characterize. So whatever we do post-harvest, we are completely with the understanding of what the profile looks like. So here and then some of the studies we did, in fact, we grew the seedlings at different temperatures. Then the higher temperature, which is supra-optimal, it somehow triggers the high level of antioxidant. That antioxidant, the pool, then actually contributes the total antioxidant capacity. The epitase radical, the lowest is 3520, and in the FRAP, the higher reading absorbance, that simplifies the high antioxidant activity. Then we looked at the proline as well as the free phenols, and then the higher temperature actually has spiked the production of proline amino acid in response to higher temperature, which is drought. And then prolines their function is actually osmoregulation. It adjusts this osmoregulations. So this is actually proves in higher temperature in these seedlings. So same applies with the phenolics. At higher temperature, there was a higher accumulation of the phenolics. So then we took that and then we want to use it, for post-harvest treatments. Then, of course, with the post-harvest biology and technology, so we did with already matured fruit, and then maturity index, and then the right time to harvest, and then the fruit has to be maintained because it's harvested. To maintain, it has to go through a number of practices. Biological processes must be understood, and then accordingly, the technologies must be applied. That's optimum towards the biological processes. So you can see with the harvest over pack houses at different levels, then you end up probably not having it. So the majority dominantly wasted. In order to maintain, to minimize, we need to really approach with the pre-storage treatments, 1 MCP for blocking ethylene, which is ripening hormone. Edible coating, where actually normally it has something to do with the biology of the fruit, which is transpiration. And then post-harvest condition, the way you produce the cultural practices before harvest. The cold storage in modified atmosphere, which is manipulating the gases, oxygen and carbon dioxide. And then also the hot water treatment basically stimulates heat shock protein. They are also known for the secondary metabolite. Then essential oils, then it could be any fragrance smells in the fruits. They are because of the essential oils. We can actually recycle it, treat the fruits because it has antimicrobial properties. So eventually, we are trying to maintain the fruit quality, extend the shelf life. Of course, that also contributes to minimize the food insecurity. So speaking of edible coatings, so it's better to understand also there are a number of different sources, polysaccharides, the pectins, and lipids. So we can still this extracted from plant byproducts. So we can actually use them as edible coatings, then because they are polymers, then structure formed. Then what we did in our work, actually, we did plant extracts, mainly secondary metabolites, and then infuse it with the edible coatings. Infuse it in many ways. So that's what happens. You form a product at different percentage, then we treat the fruits in order to be able to tackle some of the deterioration factors. So in other words, maintaining the fruits during the storage as well as the shelf life. This is basically, that's what we exercise in daily activities in the post-service biology technology modules. So the benefits, we always tap on the organic and natural because the quality preservation, the reduced food waste, the food safety, sustainability. The critical thing here that our fruit industries are export oriented. So anything to do with the synthetic is a problem, with the residual. So we have to come up, with alternative natural byproducts. Those byproducts they're accessible. So what we're trying to do, we need to rethink and then approach it differently in order to make them more valuable and then also applied. Then that's also interesting for the future sustainable agriculture. So this is the plant material we use, harvested, which is actually the leaf part and seed. And then also that, finally we form the extract, and this is the part we use it for. So then one of the studies we did, we used chitosan as an edible coating, and then carboxymethyl cellulose, which is a cellulose edible coating. So it doesn't have any toxicity concern. So it is the best fit for post-harvest treatment. So this is a funded project by Department of Science Technology, as well as South African Avocado Growers Association. So what we found, this is simply demonstrating the extract performed very well. This is the same fruit, but look, the control is already deteriorating and then over-ripening, but with extract actually. Then we did the fruit mass loss. The treated ones are better compared to the control. This is a work actually published in Food Packaging and Shelf Life, and then also the second highly cited work so far in the Google Scholar. Then we have the texture-wise. So the texture-wise, firmness is highly correlated to many of the physical attributes. This is also contributed by a number of the treatments, which is the moringa extract, as well as the edible coatings. So we checked it at different cultivars, different foods, and then also funded by the South African Avocado Growers Association. Lipid peroxidation is very important for the industry because one of the problems we have in the industry is the chilling injury. So lipid peroxidation indicates the susceptibility to those storage conditions. So we also adopted ozone and UVC because we thought, this is naturally produced, and then also that we have the local manufacturing. So we can easily get it from the local markets, and then we use them for post-harvest treatments, mainly for antimicrobial properties. And then remember, one of the things that, when you apply ozone, UVC, somehow we're stimulating the biological spiking of radicals, so that also passively stimulates the production of antioxidants. So these are the industry problems, ethylene, uneven ripening in avocados, and then also anthracnose and then stem end rots is a big problem, huge loss for the industries, and then also the chilling injury. So one of the things we do, in fact, we use technology. The first thing we go for is understanding how it works. Then, of course, we follow. Then we look at how it's produced. The residue for ozone is oxygen. In fact, it's oxygen then produces with this the corona produces the O3, and then that lasts only for certain minutes. The residue will be oxygen. So the other also activity with ozone, it can also disseminate or destroys the ethylene C2H4 to carbon dioxide and water. So these are the approach oxidizers, and then this also can be integrated in the cold storage practices. You have edible coatings applied in the food, then still you can integrate also the ozone as well as the UVC treatment. So here, what I want to highlight, the effect of ozone on the level of ethylene level. So you can see ozonated storage. Of course, that it has a significant effect on the level of ethylene, which is so. This is the study we did with the edible coatings and control, but over different times. Then, of course, this is also the published work. This is some of our edible coating treated foods, and then you can see glossy, and then they perform very well. And then we have basically a product, but what is now is actually we are in the process of commercializing it. And then, of course, that we need to also involve commercial partners. So this is the work with my student on the kiwi fruit. And then this is also during COVID time, busy in the lab, working on the kiwis. And then blueberries, strawberries, citrus, and then this is also our byproduct, which is in the pack house, in the industry, where we apply the fruits with the edible coatings. So the biofungicides here, basically the sources, plant materials, and then we have the extracts, which is terpenes, alkaloids, and phenols. Go back to the pathways. Then you have a biofungicides. Basically, it's a reduced environment, minimal impact on non-target organisms, improved soil health, lower greenhouse gas emissions, increased food safety, and the reduced risk of pesticide resistance. So here with prochlorides, this was used by fruit industries, but this one's actually is going to be banned or expired in this year. So the industries are rushing to have a replacement for this one. So we have a candidate, and then we know that what is confirmed it's working. So hopefully this will be potentially the answer for this challenge. So the efficacy, of course, this is, I'm not going to more, but I will highlight a few points. Isolation, culture, identification, pathogenicity test, in vitro. This is also with the group in plant. Then we need to understand what are the entry-level method of penetrations, direct penetrations through the natural openings, and then also wounds. This is a mycelium infestation. And then after culturing, we need to identify the strains through the morphological structures. You can see collected trichum, alternaria. And then also looking at the pathogenicity test. Then we have very economic strains here, Plasmodium, and then collected trichum. And then with the extracts in vitro test, you can see that, the methanol and ethanol, which is actually the plant extract, is suppressed. The mycelium grows. And then also what happens here, and then with moringa extract, is something to do with the hyphae structures. So you can see some fragments because of the moringa extracts, which is as compared to the control. So this resulted with the suppression of mycelium growth. So with this table, we have plant tissues with a collectotrichum. Then the mycelium growth, if you look at it, the leaf extract had very significant effect when it comes to the mycelium growth compared to the control. So the nanotechnology, we believe that, that is something that, we need to do intensively, and then that will solve. So the study of nanoscale scale, which is 1 to 100 nanometers. So the advantage is efficacy, controlled release, stability, physical properties, reduced environmental impact, and better uptake. So here in agriculture, it can actually be used for many things, for biofertilizers, and polymers, you name it, in different ways, and then also can use. So with synthesis, these are the characterization we need to adopt. Once you synthesize, is it the heating oven approach or the microwave synthesis or the green synthesis? It has to be confirmed through these processes. And then for morphology structure, then the TEM, which is transmission electron microscopy, scanning electron microscopy, and then all this has to be in place. Then the size has to be between 1 to 100 nanometers. Your bigger surface area to volume ratio. Then how it actually reacts, which is by rows, and then by nanoparticles, then basically it acts directly to the. So we have some research outputs we did publish on this work. And then this is what we synthesized from zinc salvate, zinc oxide nanoparticles. This is actually the product which I said earlier is ready for potential use. So we can upscale it. It's not a problem. Then this is actually, we believe that is a game changer. Then we're going to really benefit more from here. So this is the characterization we did, SEM, the TEM, and then also we need to check also whether it's through EDX, whether the zinc is already present, and then the FTIR, which is actually whether the with the mix, actually the integration was as the leaf extracts present structurally. And then this is the shape file, X-ray diffraction spectrum. And then here we tested it in the mycelium growth, and then that showed that there was significant impacts on the pathogens treated with the nanoparticles as compared to control. Here, I think with my team that we were conducting pack house trials. In order to register, you need to do it at the pack house. So we are grateful for the Westphalia, where they allowed us to do the treatments in the pack houses. So we are saying that this is plant-based materials. It minimizes the usage of harmful and toxic materials, which is also a guarantee for our export foods. Moringa-based also zinc oxides can be recommended also as environmentally friendly substitute for prochloride. Then the future, in my thinking, of course, many of us, nanotechnology is the way to go. And then we also mix it with the plant-based agriculture inputs, fertilizers, biofuels, pesticides, and then also others. Then the waste, we can change it to valorization. There is an opportunity. I'm sure it doesn't have any limitation to do that, but we need to find also the right optimal extraction protocol. And then also characterizing also still ongoing secondary metabolites, as I mentioned, there are more than 50,000 so far, but still. So these are the people that work in many ways. I'm very much grateful. Then this is a postgraduate team. That's most of them are colleagues. They really contributed in this moringa research and then antioxidant. I'm grateful for the support. And then this is also my engagement in West Africa, where I did contribute the post-harvest research and then also teaching and representing the University of KwaZulu-Natal. And then here, engaging with the farmers, I always believe with the farmers, and then there should be impact. Whatever we do, research has to go far. We don't need to put it in on the shelf. It has to go down to the ground, and they should change lives. So that's my strong recommendation. So we have knowledge, we can do more, but we need to be impactful by changing lives. So we have resources, so let's do it. So with that, this is my university, long time. This is where I come from, University of Asmara. I'm grateful for everything, and then thank you. Thank you so much.
Thank you so much, Professor Tesfay, for a wonderful presentation. I think all of us here will agree that indeed at UKZN we inspire greatness. And that's how we build our professorate. And we're so proud of you on behalf of the Vice Chancellor and the Executive Management and Senate. We are truly, truly so proud of you and your achievements. And we hope that we're gonna, soon, we are the leaders in the post-harvest, and we are in a very competitive space, SNR, and it's quite very good to build physiology and biochemistry together to become an applied horticulturist. We're so proud of you, and we want to wish you all the best, and thank you very much. Thank you, colleagues.
Thank you.
Congratulations, Tesfay. Well done.
Thank you, Prof. Thank you, Prof.
We really enjoyed your talk.
Okay. Thank you, Prof. Thank you. All right.